Welcome to part 3 of 3, looking at chapter 12. In this chapter, we've been looking at gene expression at the molecular level. So in the first part of this chapter, we kind of talked about the general process, which again I'm showing up here in prokaryotic and eukaryotic cells. And then part 2, we talked about our transcription part, so how we took our DNA and we transcribed it into an RNA copy. This last section, we're going to talk about the RNA processing that occurs in our eukaryotic cells, and then we'll also talk about the translation process, so how we take that RNA copy and we use it to create our polypeptide in the ribosome. So first off, we're going to look at the RNA processing in eukaryotic cells, which doesn't happen in our prokaryotic cells. So this RNA processing, bacterial mRNAs, they can be translated into polypeptides as soon as they're made. However, eukaryotic mRNAs, when they're transcribed, they are called pre-mRNAs. Those pre-mRNAs, they require processing that will turn them into mature mRNAs. And then those mature mRNAs, they can go through the translation to create the polypeptides. So the three types of processing that occur, we're going to have splicing, where we're going to remove parts of our mRNA called introns, and then we'll connect another part of our mRNAs called exons. The other thing is that we can put poly A tails on the 3' end of the molecule, and then on the 5' end, we're going to put a 5' cap on it. And the tail and the cap, they just help to stabilize the mRNA molecule and to signal that it's ready to exit out of the nucleus into the cytosol. So the splicing part, up at the top we have our DNA, where our gene is found. And again, we have our promoter on the left, we have our structural gene, and then the terminator on the right. So these structural genes are actually created of different parts called exons and introns. The exons and introns are both transcribed into our pre-mRNAs. Once that pre-mRNA is created, then the splicing can occur. So we're going to cut out our introns, and we're going to leave the exons, and we'll splice those exons together in this example. And then in addition, on the 5' end, we have the little 5' cap, that's the light green. On the 3' end of our pre-mRNA, we'll put a poly A tail. So the splicing part, introns, we have them in a lot of eukaryotic genes. They're kind of like extra pieces of information that we don't need in order to create our polypeptide. So we have special enzymes called spliceosomes that are going to remove those introns. And these spliceosomes, they're composed of subunits called SNRPs, which are small nuclear RNA molecules. And the neat thing about eukaryotic organisms is that you can have something called alternative splicing, and this is where in some cases you can have like 3 introns that need to be cut out, but in other cases in that same RNA molecule, you may only have 1 intron that needs to be cut out. So you can actually create different products off of the same gene. So alternative splicing, you can splicing can occur in more than one way to produce many different products from our one pre-mRNA molecule. The splicing only happens on mRNA molecules. rRNA and tRNA, they are capable of self-splicing, so they're also called ribozymes. So the splicing, again, only occurs on our mRNA molecule. So this is kind of how it occurs. So up at the top we have our pre-mRNA molecule. The exons are in yellow, the introns are in green. Remember, we want to cut out our introns. So what happens is that at the beginning of the intron, we have a SNRP attached. In the middle of the intron, we have another SNRP subunit attached, which is on that branch site, the little black section. And then the end of the intron, you'll also have another SNRP. The SNRPs on the beginning and the end, they move towards each other. Your intron kind of folds up, So the snurp in the middle kind of pulls it up. And then your two snurps on the beginning and the end come together. They're going to cut that intron out and just lift it out of your pre-mRNA molecule. So it'll look like this. So you can see the beginning of your intron was cut and attached to the branch site, and then the end of your intron was cut, and it's just lifted out of our pre-mRNA. The two exons that were next to each other, they're going to be connected together. So besides the splicing, you'll also have this capping that occurs on the 5' end. So we're going to have a modified guanosine attached to our 5' end. And this cap, it's needed in order to allow that mature mRNA to exit out of the nucleus into the cytosol, and it also helps with your mRNA to bind onto the ribosome during the translation process. The poly A tail on the 3' end, we're going to have 100 to 200 adenine nucleotides added on, so just a bunch of A's added onto the 3' end. This is going to help that molecule last longer. It increases the stability and lifespan in the cytosol, so it's not broken down as quickly. And then your mRNA can go through translation. So here's kind of a summary of, up at the top we have our 5' cap, that modified guanosine cap on the 5'. On the 3' we have that poly A tail that's added on. Okay, so just to point out kind of where we are at this point, we talked about transcription, which is very similar between prokaryotic and eukaryotic cells. We also just went through RNA processing that occurs in the eukaryotic cells. So now we have mature mRNA molecules that are ready to go through the last part, which is going to be the translation part up here. So now we're going to take that mRNA, we're going to bring in a bunch of players that are going to help and turn that information into a polypeptide, which will then become a protein, which will then give us all of our traits and characteristics. So this translation part, in order to do translation, we need something called the genetic code. So a sequence of bases in your mRNA molecule, we're going to read those bases in groups of 3 called codons. So an example of a codon would be like AUG. So 3 nucleotides in a row, that is a codon. And this genetic code is degenerate, so more than one codon can specify for the same amino acid. So because we have 4 different nucleotides, we read them in groups of 3. There's 64 different combinations of codons that we can have in our RNA molecule. But remember, there's only 20 different amino acids. So some amino acids are coded for by 4 codons, there's a few coded for by 6 codons, and on the next slide we're going to see this genetic code. So they have actually figured out every codon and what it codes for. So here is this genetic code. It's table 12.1 in your book, and you will have to be able to read this. Okay. So what happens is, for example, I mentioned AUG as a codon. So the A is in the first position of the codon. So first position, if you look on the left-hand side of this chart, it says first position. And we have UCAG. So if we're looking at the codon AUG, we have A in our first position. So we're going to be looking across that group. So if you look at the A, everything going to the right starts off with an A. So we're down to 16 codons going across. Our second position was a U. So if you look going across the top, it says second position, we have UCAG again. So we have U, so it'll just go down on the left-hand side in the U's. So we're down to 4 different codons, and they all have AU. So we have AUU, AUC, AUA, AUG. So right there we have our AUG. That AUG codon starts for a methionine amino acid, and it's also called the start codon. So that is a really, really important codon, the AUG. And I'll point it out again. Let's see, if we want to do another codon, if we do CAA, first position we have a C, so you'll see that's the second row. In the second position we have an A, so second position, find the A, go down into the box. We see 4 codons, they all start off with CA, 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 CA. If we find the CAA, that codon codes for the GLN amino acid right there. And the amino acids, you can just represent them as a 3-letter abbreviation. Okay. So the other thing I want to point out, let's see, if you look in the bottom left-hand corner, we have GUU, GUC, GUA, GUG. All 4 of those codons code for the same amino acid, the valine, VAL amino acid. So that again is showing that every codon codes for an amino acid. Those amino acids can be coded for by multiple codons. And then the last thing I want to point out is that the top right-hand side, you'll see that we have 3 codons that say stop next to them. Those are said to be the stop codons, and those will be important in a little bit with this. Okay, so we're going to be using that genetic chart that I just went over, so make sure you really know how to use that chart, because you will be using it on your homework and also on the exam. So the 5' end of our mRNA, we have a ribosomal binding site. So our RNA is going to be read going from 5 to 3. So we have a binding site, our ribosome is going to attach, and then your ribosome is going to start to travel 5 to 3, and it's going to look for a start codon, which is that AUG codon. So always, always, always find the AUG. Don't just start with the first RNA codon that you have. Look for the AUG. And then at the end of your mRNA molecule, towards the end, there's going to be one of 3 stop codons, and I pointed those out on the chart. This is where we're going to have termination, so we're going to stop the translation process. So we're going to have a start codon and a stop codon, so it tells you where to start and where to stop. And then in between that, that's what we're going to translate. We're going to use those codons in between the start and the stop to tell us which amino acids we should hook together when we create our polypeptide. Alright, so this part that we're going to translate is called the reading frame, and again, I don't know why this is something that students don't get, but you don't just start at the start of your mRNA molecule. You have to start the 5' and then read down until you find your start codon, because we have to have a start codon in order to actually start the translation process. So this example here, we have at the 5' end, we start looking, reading towards the right, and you just keep on traveling down until you find AUG. That AUG codes for the start amino acid, which is our MET. And then from there you can start grouping your letters into groups of 3, into the codons, and then those codons you'll use your chart to figure out which amino acid is going to be hooked on next. So in addition to our mRNA that we have talked a lot about, we have those codons on there, those set of 3 RNA nucleotides. Those codons are going to match up with a tRNA molecule that has an anticodon on it. And these are a 3 nucleotide sequence. They're part of a tRNA molecule, and it's going to be complementary to the codon. So codons and anticodons, they will match up with each other using those base pairing rules that we've talked a lot about. In addition to that, we have a ribosome, which I have kind of showed you a little bit about. So we're going to look at these key players, and then we're going to put them all together to do the translation process. So again, we talked about the mRNA a lot, so you should know about that by now. The tRNA, I kept on mentioning we're going to get to it, so here we are. So tRNA, there's different tRNA molecules that are coded for by different genes, and tRNAs, there's 64 different tRNAs. So each tRNA has a specific anticodon on it that matches up with one of those 64 codons in our chart. And then these tRNA molecules, they're transfer molecules. So on one end, they have their anticodon, and then on the other end of the tRNA, that's where our amino acid is going to be carried. So tRNAs, they're the ones that are going to use that chart to figure out which amino acid is going to be next. There are lots of different tRNAs. The common features, though, they have what's called a cloverleaf structure to them. They all have their anticodon on there. And then the other end, they have that acceptor stem where the amino acid is located. In addition to the tRNAs, the process where this all occurs is the ribosome. So prokaryotic organisms, they have one type of ribosome. Eukaryotics, they have different distinct ribosomes in different parts of the cell, which makes sense because eukaryotic cells are always more complex. But basically, ribosomes, they have a large subunit and a small subunit. And those two subunits, they will come together during this translation process to help bring everything together. So the ribosomal shape is determined by rRNA. So if you remember, we have mRNA, we have tRNA. The rRNA is the ribosomal RNA, which is found in the ribosome. And these ribosomes, they have 3 discrete sites in them. These discrete sites are for tRNA binding and polypeptide synthesis. So on the right-hand side, we have an A site. This is where our tRNAs are going to enter in, and the anticodons match up with the codons. The P site, this is where our polypeptide is located, so it's coming out the top, those blue circles, which are the amino acids all hooked together. And then on the left-hand side, we have the E site, which is the exit site. This is where our tRNA is going to exit out of the ribosome. So translation occurs in 3 stages, and luckily they have the same name as the transcription. We have initiation, where everything comes together, elongation, where our polypeptide is created, and then termination, where we get to a stop codon and things will dissociate from each other. This slide is showing the initiation part. So the top left-hand side, we have our small ribosomal subunit in the orange. That ribosomal subunit binds onto the blue ribosomal binding sequence on our mRNA. Okay. And then once that is bound on, we have our start codon in the green, the AUG located in the P site, right there. So this AUG, remember that's our start codon. Always, always, always look for the start codon. That is where you're going to start translation. So this is where our first tRNA comes in. So our codon is AUG. The anticodon is UAC because we use that complementary base pairing. On the other end of our first tRNA, we have our first amino acid, which is that blue circle up at the top. Okay, so we have our small ribosomal subunit attached. The mRNA is in there in the yellow, and we have our first tRNA with our first amino acid. Once that is all set up, the large subunit is going to attach on to the small subunit, and then that is when our initiation is complete. And this is shown on the right-hand side, our picture on the right first. So we have our A site is open for our next tRNA to enter into the ribosome. our first amino acid, our starting polypeptide, is in the P site, and the E site right now is empty over here. So we have our initiation. Everything is put together. We have our mRNA, our tRNAs, and our ribosome all attached to each other. So now we're all set up. We can do translation, we can do the elongation, we can create our polypeptide. So this elongation process, how it occurs, so our next tRNA, it's going to enter into the A site, which is open, and it's bringing the next amino acid. So our next tRNA enters into the A site. Our previous tRNA is in the P site, and it's holding that polypeptide that's shown in the blue, and it's coming out the top of the ribosome. So our next tRNA enters into the A site. So we're reading the codon CGU on our mRNA. The anticodon on the tRNA is GCA. So remember, they're complementary to each other. And then on the other side of the tRNA, we have our amino acid, which is in the yellow at the top of this. Okay. So our next tRNA enters into the A site. Then what happens is our polypeptide chain, it's going to form a peptide bond between that new amino acid that just came in the yellow and your growing polypeptide chain. So that blue polypeptide chain attaches on to the new yellow amino acid that just came in. And this is called the peptidyl transfer reaction. So we're adding on our next amino acid. Then what happens is that our ribosome is going to move over one codon to the left towards the 3' end, because remember we're going 5 to 3. So our tRNA that was holding the polypeptide goes into the E site, which is the exit site, so it's going to exit out of the ribosome. Our tRNA that's holding the polypeptide is now in the P site, and that opens up the A site. So our next codon can be read, which is GCU, which is in the A site. The anticodon on the incoming tRNA is CGA, and then on the other side of our tRNA that's just coming in, we have our next amino acid. And then this process just happens over and over. We read one codon at a time as our ribosome moves down the mRNA. So that's our elongation process. We just add on one amino acid at a time based on the next codon. And then we have our last step, the termination process. So termination is when we reach one of our 3 stop codons when it is in the A site. This is when translation ends. So remember, the 3 stop codons based on our chart are UAA, UAG, or UGA. So instead of a tRNA entering into the A site, we have another factor called a release factor that enters in when we get to one of these 3 codons. So what happens is that we got to a stop codon, which is in the red on our mRNA, so UAA. Instead of a tRNA entering in, we have a release factor that enters into the A site on your ribosome. This signals that translation is done. The tRNA lets go of our polypeptide. So the tRNA disassociates from the polypeptide, which is released. And then once that occurs, our ribosome subunits release the mRNA molecule. So now we have completed this translation process. So here we have a summary again of the central dogma of molecular biology. So we went through kind of the general ideas and the research behind the discovery of the central dogma in the first section. The second section we looked at transcription, so taking our DNA, using it to create an RNA molecule. And then in this last section we looked at the RNA processing that only occurs in eukaryotic cells. And then we also looked at the translation process up here. In your homework and lab and on the exam, you're going to have to take a DNA molecule and you'll take it through transcription and also translation. So you're going to have to actually do this process and use the complementary base pairing rules and be able to read that genetic code chart. So make sure that you can figure that out and you know how to do that process.